Greetings, dear students. Let us learn about the different important concepts involved in the technique called plant tissue culture. So basically, plant tissue culture is described as the technique of growing cells, tissues or organs isolated from parent plants in an artificial medium under in vitro aseptic conditions. The whole concept of plant tissue culture originates from the ability of an individual plant cell to regenerate into a complete or whole plant. This concept was proposed by Haberlandt way back in 1902, and ever since the field of plant tissue culture has grown by leaps and bounds. So he said that due to the intrinsic ability of isolated plant cells for autonomous life, they would be able to regenerate and give rise to a complete plant. The concept of totipotency is observed in animal cells also, but due to various differences that exist between the structural organization of cells and tissues between plants and animals, the totipotency and regeneration ability are observed only in plant cells, because of which they can grow into individual complete new plants. So a totipotent cell is a cell, generally an embryonic cell or a stem cell, which can give rise to all other types of cells that are seen in an organism, as well as the totipotent cell can give rise to cells of its own kind. Pluripotent cells are limited in their ability of giving rise to different types of cells. Multipotent cells can give rise to a few other type of cells, while these multipotent cells on maturation become differentiated and specialized, giving rise to somatic cells. So this concept exists both in plants and animals, but we can see the regeneration only in plant cells. So basically in plant cells, to utilize this property of totipotency, a small tissue or part of the parent plant is collected and then incubated under laboratory conditions, during which it may give rise to a callus, it may give rise to adventitious vegetative buds, or it may give rise to structures called as somatic embryos, which resemble in structure and function the zygotic embryos seen in the seeds. There are several important terms you need to understand before going into the details of plant tissue culture. Let us begin with the explant. Explant is the initial material or the starting material for a tissue culture process. The explants that are generally used for plant tissue culture are leaf, shoot tip, root tip, meristem tip, as well as nodes. Many times floral organs or reproductive organs like the ovule, ovary, anthers, all these can also be used as the explants. So explant is the initiating material for a tissue culture process. Cell differentiation is the process by which cells either in the root, shoot or apical meristems or in the cambium mature to give rise to specialized cells performing specific functions like the xylem cells and the phloem cells. In this process of maturation or differentiation, the cell undergoes a lot of structural changes. For example, the protoplasm is lost during the formation of tracheary elements. Dedifferentiation is the transformation of plant cells from a given differentiated state to a less differentiated state, which means from a mature state, a cell reverts back to a stem cell like state or an embryonic state. Stem cell related genes play important roles in dedifferentiation. Redifferentiation is a process in which a dedifferentiated plant cell like the callus cell once again loses its capacity to divide and give rise to different types of cells. Instead, it becomes mature and specialized in its structure and function. A plant callus is a growing mass of unorganized or undifferentiated plant cells. In living plants in situ, that is plants growing in field conditions or in pots, callus cells are those cells which are formed during the healing of a plant wound. Media is a synthetic nutritional substance that is provided to plants under culture conditions. So there are different components that are added to give rise to a media, which we are going to learn in the next few video sessions. In vitro conditions refer to controlled laboratory conditions. Aseptic technique is a method in which microbial contamination is minimized to prevent growth of unwanted microorganisms in the cultures. Plant tissue culture generally occurs in the following sequence of steps. The process begins with the preparation of media, during which different components of the media are weighed and mixed to give rise to a defined media. Parallelly, explants are collected from the chosen parent plant. The explant is of different types based on the species of the plant that is under consideration. The collected explants are then subjected to a process of disinfection called as surface sterilization, during which they are treated with different chemicals to remove the microbial load over the surface. The disinfected explants are then inoculated onto synthetic nutrient media by following aseptic conditions or aseptic techniques. This is a process in which we take care to allow minimum number of microorganisms to enter the culture, so that the explant is not disturbed or destroyed by microbial growth in the culture. The inoculated explants are then incubated under in vitro conditions, where we maintain the required physical conditions like temperature, pH of the medium, and other important physical conditions which come into play in plant tissue culture are the light period and the light intensity. So the light period is also called as photoperiod. Now, once the plants or the explants establish themselves in the culture, giving rise to either a callus or adventitious buds, the cultures are subjected to subculturing. Subculturing is a process in which we divide the available culture material into portions and inoculate each of these portions into fresh media in fresh containers. This is generally done to increase the number of propagules or to induce the formation of different organs in the plant tissues. So once subculturing of calla is complete, they are allowed to undergo regeneration and organogenesis, or it may be somatic embryogenesis in some cases. Once young plantlets are developed in the culture containers, they are subjected to a process called as acclimatization, during which they are allowed to adjust to environmental conditions found in the field conditions. Once acclimatization is done, the plants are then transplanted into field or potted conditions for further growth. So this is a pictorial representation of the general steps followed during tissue culture. So from the adult plant, we choose the explant, then isolate a part of it, subject it to disinfection, and then inoculate it onto synthetic nutrient media. When incubated for a certain period of time, which is generally 3 to 4 weeks for most of the plants, it most of the times give rise to a callus. In some cases it may be adventitious buds, in some plants it may be somatic embryos. So the callus then gives rise to organogenesis, that is formation of the shoots and organs, or the callus may be also used to initiate a suspension culture from which then shoots and roots are induced. So either way it ends with organogenesis. The newly developed plantlets, young plantlets, are then subjected to hardening or acclimatization followed by transplantation. Based on the type of explant and the mode of regeneration, there are different types of plant tissue cultures, which may be callus culture, cell culture, embryo culture, when embryo is being used as the explant, seed culture, wherein the purpose is to give rise to aseptic plantlets. The plant tissue culture may be meristem culture if meristems are being used as the explants, protoplast culture, or organ culture. Organ culture includes the use of reproductive organs like embryos, ovules, ovaries, and anthers, or it may be even the meristems for regenerating under different conditions. So these are all the different types of plant tissue culture. The regeneration potential of these different organs and explants varies between the plant species, and even within the same species it varies between the subspecies, varieties, cultivars, or ecotypes. So based on all these aspects we choose the suitable explant. Once an explant is chosen, the explant may undergo different pathways of regeneration. Most common pathways of regeneration are micropropagation, in which the chosen explant gives rise to a callus. From the callus, shoot induction and root induction are done. Some of the times when organs like embryos and anthers are used, again it may be via callus or directly without the formation of callus. In certain conditions, either from the callus or from the explant, somatic embryogenesis may be induced. We will learn about the details of all these pathways in the next few video sessions. Let us have a brief look at the applications of plant tissue culture. Maintenance of pathogen-free plants. So whenever a plant gets infected with microorganisms like bacteria, fungi, especially viruses, it becomes difficult to propagate from that plant as the starting material, because generally the entire plant body carries the infecting organism. Particularly in case of viruses, the whole plant gets infected. Under such conditions, if the infected plant has to be used as the starting material or the parent plant, tissue culture is the only methodology that can be used. So the meristem tips of any plant are devoid of viruses as well as other pathogens. So by collecting the meristem tip from the infected plant and initiating the tissue culture, we can produce pathogen-free propagules. Then rapid clonal propagation is another important application of plant tissue culture. Clones are individuals which are identical to each other. So when we can produce plantlets which are identical with each other in terms of growth period and all other aspects, it becomes easy for the farmer to cultivate and harvest the crop. Also, in certain plants where seed propagation is not possible or seed propagation involves a lot of variations, tissue culture is the only available method. Many horticultural crops are being propagated by the micropropagation or clonal propagation technique. For example, banana, teak, grape, and so on. Several floricultural crops are also, like orchids, are also propagated by clonal propagation methods. Production of transgenic plants which have unique genes or foreign genes is a transgenic plant regeneration. So these transgenic plants generally will not be able to grow into complete plantlets without the tissue culture methods. Plants are known to produce thousands of secondary metabolites. These secondary metabolites have huge number of applications for mankind. They are used as perfumes, essential oils, flavoring agents, medicinal substances, and so on. So the production of these secondary metabolites from plant tissues and cells using suspension cell cultures is an economic and highly useful way of producing these substances. Tissue culture methods are also useful in understanding the basic biological processes like morphogenesis, cell and tissue morphogenesis, differentiation, cell division, metabolism, radiobiology, so on. Plant tissue culture is highly useful in the production of haploids. Haploids cannot be produced easily by natural methods. So the advantage of these haploids is they allow the expression of recessive characters in the plants. So if any recessive characters which are desirable are identified among these haploids, then those haploids are subjected to chemical doubling of chromosomes, and such plants are then used for cultivation. So use of tissue culture and haploid production allows production of new cultivars. Tissue culture is the only method for regenerating plants that have been produced by distant hybridization, that is hybridization or crossing between different genuses, different genera. Then it is also the only method for regenerating plants that are produced by somatic hybridization. So these are all the several applications of plant tissue culture. You can refer to these websites for deeper insights into the concepts of plant tissue culture. Thank you.